Hi everyone, just 1 second. I'm trying to get my slides pulled up and they're not cooperating. Okay. I think we're good to go. Does anyone have any questions that they want to ask before we get going today?
Jordan, I might have a question, but I need to check the status of my Slurm job.
Okay.
Can I ask you at the end of the class?
Yeah. Yeah, I'll
Thank you.
take some time at the end and we can do that.
Okay, thank you.
Yeah. Okay. Well, let's go ahead and get started then. Let's see. Okay. So on Wednesday, we went over an introduction to R and kind of bridging that divide between Python and R, what's different, what's the same. So now we want to get into more of using the actual software that's available within R. So the software that I'm going to demonstrate today, it's called DE-seq2. So this is used in RNA sequencing for determining differential expression of a gene between two conditions. And so I'll talk about why we use a software tool like this, especially when we're calculating p-values for these gene expression values, and why we can't just use like a student's t-test for doing something like this. We'll also talk about some basic data normalization. So when you're processing RNA-seq, there's some inherent biases that you're going to have to deal with just due to the technical nature of the assay. And so we'll talk a little bit about that, as well as how to generate a basic scatter plot to compare two biological replicates to ensure that they are, in fact, biological replicates, that they are similar to one another, and they don't have some weird differentiation between the two samples. So I will basically, what I'm going to do is just kind of walk through the steps of using DE-seq for basic analysis. And I'll have the actual commands that I used where you can kind of highlight and copy and paste. So just walking through this. So we're working in that interactive console region of RStudio. And so the first step is importing the DE-seq library. So loading all those DE-seq functions into memory so that we can use them for data processing. And as you can see here, so when I ran, I think this is before this command, I ran that !, DE-seq2. It pops open the help menu that you can scroll through if you want help using a specific function or need to know what arguments are available for a function. You can find that out from that information there. So when we're dealing with data for DE-seq, we need two different inputs. And the first one is the count table that we generated during the sequencing alignment. So those are integer numbers for each of the genes. So here you can see I imported that count table. And as a note, so if you want an example data set to play with on the GitHub, repository in the folder for this lesson, I have a count table and then a metadata table available so you can play directly with that. So this is similar to your basic data frame import, just like in Python, where you need to specify the column delimiter, whether or not there's a header, what column the row names are found in. So in this case, that first column is where the G names were. So I'm going to say row names equals 1. So here you can see that I ran the head function. So this is going to print out the first 5 rows of this data table. And so you can see the format where we have the different samples as columns, the row is genes, and then you can see the values are integer values. So when you're, okay, I got a Zoom attendees are waiting email, very delayed. So just want to make sure that there wasn't some other session going on. So we'll see this, I think, in one of the normalized data frames that we'll talk about later. Kind of a good indicator of whether or not the data frame's been normalized already or not is whether or not all the numbers are integers or not. So if you see any decimal places, that's not the kind of data table that this will accept as input. And I guess the reason why you don't need to pass in a pre-normalized table to DESeq is that DESeq will handle the any normalization that needs to take place under the hood. So the second data input is a metadata table. So, and we'll open up an example of exactly what this looks like. But again, we're reading this in as a tab separated table. To be perfectly honest, I don't know why I always do this text equals read lines command. I think as long as it's a TSV formatted or text formatted table, you should be able to read it as above. I think this is just one of those things where this is how I did it the first time, and generally I just copy the code, modify what I need to, and so this is kind of carried over through the last couple of years. But you'll see that, I guess one distinction is here, you're not going to, it's actually interesting. So I don't indicate the row name column. So I just read in the row names as a column itself with the sample names. And it'll just give it a default column name of X, since there is no column header for that column. And again, we can run the head command on the sample table and view what that input looks like. So just to take a closer look, so this is just that count table that I got from aggregating the counts from htseq on the different samples. And then here's my metadata table. And so as you can see, this kind of has all the information for my sequencing run for the different samples. So I have like library input concentrations, volume of input for the library, QC numbers before it was run on sequencing. So all this stuff that isn't really relevant to the DESeq run is there as well. But you can just keep that one central metadata table, so you have all that other information. But we're going to be focusing on where the comparison is going to come down to is we're comparing the genotypes. And so you also notice that I have wild type and MCT1 deletion here, but in front of each of the wild types, I put an A. The reason I did that is by default, so you can, if you're through the documentation, it'll tell you how you can not have to write this in alphabetical order, but just for simplicity's sake here. So when it's comparing two conditions, it's going to take the denominator first alphabetically. So if we want to compare MCT over wild type, that denominator wild type is going to be whatever alphabetically first. So since wild type is W is not before M, I add the A there before each of the samples, just so that is clear. Okay, a question, is the sample table the same as the metadata table that we download when doing the SRA files from GSC set? So it's very similar. So this one isn't exactly from the SRA file data table, but with data sets that I've pulled from Geo, I've usually used that SRA metadata table. And I maybe have to, like, there'll be some cases where maybe there's two files for a single sample. And so you may need to clean up some rows so that you're just comparing basically one row per sample. And you may need to do some modification like the genotype name to make sure it's considered alphabetically. But for the most part, the sample data table is very similar to the SRA data table. Okay. And then just for reference, again, here's the commands that you can copy and paste. And I hope that all of these special characters, okay, there's some weird special characters there. So just be aware if you are going to copy and paste directly. I think most of them should be fine, but like there's this weird quote symbol that got copied weird that you may need to fix. So just be aware of that if you are just copy and pasting. You'll probably run into an issue with input formatting. So now what we're going to do is take that input data and actually process it using DESeq. And so the first function that we're going to use is this DESeq data set from matrix. So it's basically going to read the count data and the sample data into a single variable, like an object that DESeq can then use, because it's expecting a specific data format. So you can see here I guess, three required inputs. There's the count data, which is our count table, column data, which is our sample table, and then the third one that we're going to do is, this is how you actually want to use this command. You can do as I have it written here, but this is just kind of a forcing the formatting of the input, which isn't necessary if you're manually typing it in. But here what we're doing is basically setting up a similar to like a linear regression formula if you've used other statistical software before. But in this case, since we're just dealing with one condition, which is genotype, all we need is that input is this design equals tilde genotype. But in other cases where maybe you're dealing with multiple conditions, so maybe, I guess, kind of the example that I'm most familiar with would be ribosome profiling, where if you're calculating translation efficiency, you're comparing two different conditions, but then you're also comparing the ribosome footprints over total RNA to get that translation efficiency. And so, and if you read through the documentation for DESeq, you'll find more information about how to exactly set up this, but you can basically consider several variables. Maybe if you're dealing with human data, and maybe it's like lung tumor samples versus normal tissue samples, but then you also want to control for whether or not they were a smoker or whether or not they were obese. And you can set up formulas that will factor in those additional confounding variables that could be present in the population. But I would read, if you are looking into situations where you're dealing with more than one condition, I'd read that section of the documentation thoroughly. So just look at where it discusses the design argument in the documentation. So then we will actually run DESeq on our data on that object that we just created. So in this case, I am directly transforming that original variable, but if you wanted to say like DDS underscore output to keep the input and the output separate, you could do that. But then you should see this progress log as it's running through each of the steps in the DESeq analysis pipeline. And to just extract the results, you're gonna use this results function on that output. And then here what I'm doing is taking the output, I am running this order function. So basically sorting the results rows based on the p adjust column. So this will order basically from smallest to largest adjusted p-value, order the results so that they're a little bit easier to look at. But again, you could just do that step in Excel if you wanted to. And then finally, I'm going to export these sorted values to a data table, a new data table, where I'm going to indicate, again, the separation, the what value is being, or whether or not to use column names and row names. And I guess one other thing, I think this is where you need that. Actually, you don't need that. I don't think you need that data table library, but you need to convert this output data object to a data frame so they can write it to a table. And so you can just input that ordered results into this as data frame function. And you'll see as I'm going through these steps and creating new variables, you'll see here I only have those two input variables of the count data and metadata table. And now I have all these additional objects that were created while running DESeq. Okay, and again, here's that summary of commands that you can run. But we can look at the output here. And so I guess the two columns that you'll be most interested in are this log to fold change and this p adjustment value. And here you'll notice that I just have the gene IDs, but if you had inputted a count data table that you had processed using your Python script where it had the actual gene names, then you would get gene names here. Or additionally, you can just tell HTSeq to give you your counts using gene names instead of gene IDs when you're processing the initial data. And just as a note, so here when I'm inputting the data, so you'll see I'm just using that tilde desktop, my folder name. So this is what you can expect on like Mac or Windows, or sorry, Mac or Linux. On Windows, the file path will be a little bit different. I would maybe ask Ian if you run into issues specifying a file location in Windows. But I think normally, and Ian, you can correct me if I'm wrong, I'm just going to type this in the chat so people can see what this is going to look like. So it's normally going to be something like C/user/username/desktop ... But Ian, feel free to.
Yeah, that looks great.
Okay.
No, I think yeah.
Okay. So yeah, whatever your login name is for your desktop account, you would input that for username. But if you are working on Windows and you run into any issues with that, just let us know and we can help walk you through that. Okay, so now let's talk about biases in sequencing libraries and why we need to be using DESeq in the first place. So if you look at, so this is from one of my samples that I was processing earlier. So this is just one of the replicates. And you can see that if you look at basically how many times you see a given value in the count table. So down here is like, you can say these are your gene count values, and then your height here in the plot is the number of genes that had that number of counts. So you can see for the most part, most genes are not going to have any counts. And you're going to have this kind of skewed slope where you're going to have more genes are going to trend towards fewer counts, and fewer genes are going to have higher counts. And so if you look at this, you see that this is not a normally distributed data set. So using something like a t-test to calculate your p-value to determine whether or not something is differentially expressed would not be appropriate, because a t-set is assuming a normal distribution. So that's why we use something like DESeq, where it is in the underlying statistical analysis, it is assuming this negative binomial distribution. So is there any questions about this distribution or using DESeq before I continue? So let's say we are not using DESeq and we want to make our own scatter plots to compare two samples. So this is the case where pretty much anything that you're not using DESeq for or DESeq with, you're going to want to perform some kind of library normalization. And there's a couple sources of bias that we need to account for and why we're doing this. So the two main factors are sample to sample. You're going to have a different number of counts total. And so this is just a library sequencing depth issue. Maybe the concentration that was added of one sample was a little bit higher than another sample, and so that's why you have more counts from one sample versus another. Because as a reminder, when we're sequencing, we're pulling several samples onto the same sequencing lane. So you're not going to have the exact same number of counts sample to sample. So I guess the example for this is maybe you have a gene where between two samples, there's 500 counts each, but one sample has 100,000 reads, the other sample has 100 million reads. And so basically 500 out of 100 million versus 500 out of 100,000, those are going to account for different fractions of the total read amount, so different concentrations of RNA fragments. So you want to be cautious of that, because you could either say, oh, these genes are exactly the same when they're not, or maybe you have one gene has 500 counts, in another sample it has 1,000 counts. So by default, you might think, oh, this gene is expressed twice as much in one sample versus the other. But when in fact, the total number of reads for that second sample were double the first one. So the amounts are the same. So another, I guess, false positive there. So then the second source of bias here in your count numbers is going to be from the length of a transcript. So consider you're comparing. So this is, I guess, if you're only comparing one gene sample to sample, this isn't as much of an issue. But if you want to compare two genes to each other, you're going to want to normalize out this transcript length variance. So if one transcript is 10 kb, you're naturally, when you're fragmenting into these reads that we're going to sequence, so we're doing 50 base pair sequencing, so we're fragmenting the RNA into 50 base pair chunks. So a transcript that's 10 kilobases is naturally going to fragment into 10 times as many reads as a transcript that's only 1 kilobase. So there are some libraries that already exist for doing this within R. So this is one example. So I mean, if you're looking at all these samples, just the total gene counts that are present without any normalization, just based on that count table. And if we just run this box plot function on the matrix, you can see that there's some variance. And to be honest, these samples aren't too bad. The number of total reads sample to sample, there's not a huge amount of variance, but we're going to use this modified reads per million or transcripts per million method from this TWE-DE-seq library. And so you can see after you do this normalization, it accounts for some of that sample to sample variance, and you're able to normalize out some of that noise. So then we can just run the box plot on this normalized count table. And I believe there are, so this is just accounting for that sample to sample variance, but if you wanted to do sample to sample variance plus gene to gene variance based on that transcript length, there should be other libraries available that you can use where you can provide a GTF file as a reference for transcript lengths. Or again, that's another use where in Python you create a function to extract the transcript lengths per gene, make a gene to transcript length mapping dictionary, and then normalize out that transcript length from the count table. But if you want to do some of that manually, I would look at this URL that kind of breaks down the equations that build these different normalization methods and describes when one's appropriate versus the other. Okay, so now let's compare. So we took this normalized count table, and I'm running this plotting function. So this is, I guess, nice where a lot of these plotting functions are built into R. And it's as easy as just saying plot, and you're going to get a scatter plot. Before we wanted to make a box plot, so we just said box plot. And a lot of these are just going to take as a default input a count table, but then you can add X and Y labels, figure titles, format the sample names, different things like that. But here I'm looking at so column 1 and column 3 were biological replicates. And so if I plot the log transformed count columns for each of these samples against each other, I see this very tight correlation. And you can also calculate using these two columns, Pearson's correlation coefficient. And so there's a function for that is in R as well. So you can look at the strength of that correlation. And you'll notice, so here's the non-biological replicate. So most genes generally, even across biological replicates per organism, they're still going to have a decent correlation. And really what we're looking at for that differential expression is kind of these genes that are peaking up a little bit here or there. Just lost my train of thought. So within an organism, generally between two organisms, for them to be living, you're going to have a Pearson correlation coefficient of like 0.6 at least, whereas between biological replicates, it should be about 0.95 or higher. But I do want to point out here, so even between these biological replicates, you have kind of this fanning out at the bottom quadrant. So this is on this log scale. This is about anything with 100 counts or less. And this is normal. So this is just, I guess, from a probability standpoint, I guess if something is of lower abundance, like say a very lowly expressed gene, if you're just randomly sampling from all the genes present, you're going to have a lot more variance in your sampling during sequencing. And so one gene between biological replicates, it may be 5 counts in one versus 10 in another, and that's going to account for this spread out there. And actually, a lot of analyses will not analyze genes if there's less than 10 counts. And there has to be at least 10 counts per sample across that gene to be considered in the analysis. With DE-seq, that's not necessarily necessary to do that pre-trimming of low abundance genes before you input into DE-seq, because it will factor some of those low abundance genes out before it actually processes the data. So to practice this, I would take either your own gene count table and metadata table, or you can use that example one that's on GitHub. So perform differential expression analysis and identify what were the 25 most upregulated and 25 most downregulated genes that were statistically significant from your data sets. And based on the model that you use, does this make sense? Or some of these genes, were you expecting some of these based on what you know about the model? And then the second thing to do is look at some of those biological replicates and plot them out using scatter plots, and then plot out samples from different conditions, so non-replicates, and look at how those differ. So does anyone have any questions from, let's maybe start with questions from the slides from today and Monday, just in R. And then after we handle these questions, we can talk about those command line questions that were raised before. Okay. Do we want to talk about that slurm question?
Hi, Jordan.
Hey.
So I submitted a Slurm job yesterday, and I've wanted to check the status of the job, but the SQ commands that I have used are not giving me any information.
Okay. So if you run SQ and you don't see anything pop up, so SQ is only going to tell you what jobs are currently running. So if it's not showing anything, that means that the job you submitted either failed or completed. And so I would look at whatever location you output that Slurm job file. Do you remember where you put that in the supercomputer? Was that in your home directory?
I don't remember. But the thing is that